Good morning, good afternoon. This is Stephen Burley speaking, Director of RCSB Protein Data Bank. I want to take this opportunity to welcome everyone to this deep dive into computed structure model exploration using our research-focused rcsb.org website. Here's the agenda for the next hour. I will briefly introduce RCSB PDB, rcsb.org, and then our first speaker covering the first of two case studies will be Jana Rose, followed by Sebastian Bittrich. And then we've reserved 15 minutes at the end of the hour for Q&A. Please feel free to put questions into the Q&A box, and we'll take as many of those as we possibly can. The RCSB Protein Data Bank is RCSB PDB is the US data center for the Worldwide Protein Data Bank Partnership. Our research-focused website is rcsb.org, a one-stop shop for public 3D biostructure data. On this website, we provide tools for searching, accessing, visualizing, analyzing, and downloading data. The Protein Data Bank currently supports open access to more than 220,000 experimentally determined structures of biological macromolecules. And on rcsb.org, alongside those 220,000 plus experimental structures, we provide access to more than 1 million computed structure models that have been predicted using AI and machine learning methods. The provenance and reliability of both structured data types are clearly identified on our website. Now, I have one more slide to show before I introduce Jana. This is perhaps the most important slide of the entire presentation. It shows you how to opt in on the rcsb.org homepage to include computed structure models, CSMs, in the results of any searching you do. A similar toggle will appear on the advanced search page that you'll be provided access to in a few moments. So without further ado, it's a pleasure for me to introduce Dr. Jana Rose, who's scientific software developer and data architect at our UCSD site. The other two performance sites for the RCSB Protein Data Bank being Rutgers University in New Jersey, where I'm based, and UCSF. And Jana will be presenting a case study on low-density lipoprotein receptor adapter protein 1, LDL-RAP1. Jana, over to you. Thank you.
Thank you, Stephen. Welcome, everyone. Low-density lipoprotein receptor adapter protein 1, or LDL-RAP1, is a protein that helps remove low-density lipoprotein cholesterol from the bloodstream. And if this protein doesn't work well, or there isn't enough of it, the level of this bad cholesterol gets high, which leads to a plaque built up, blocking your arteries. And people with this genetic condition have so-called familial hypercholesterolemia, one of the main inherited causes of premature cardiovascular disease. So it's important protein to study. And it's actually one of the 5 proteins that make a fundamental contribution to causing this disorder. But if you look at the structural coverage of this protein, it's not uniform. Some proteins have been studied better than others from experimental point of view. So LDL-RAP1 protein is an example where experimental data provides 3D coordinates for only 5% of the full protein sequence, but there is a computed structure model with a high-level confidence prediction spanning across more than 50% of the protein sequence. And I will give a live demo. And in this live demo, I'm going to show you how rcsb.org makes it is easy to search, visualize, and access comprehensive information on protein structures and explore the additional value provided by computer structure models when applied to proteins with low experimental coverage using LDL-RAP1 protein as an example. So I'm taking you to our website. It's called rcsb.org, and an easy way to explore structures that are associated with or impacted by a specific disease is using our browse annotation feature, which gives you access to many classifications relating to structural data, including the disease ontology. So you can start typing disease term, and I am doing it live for you. Right now, you will see some suggested term. You can pick one from this list that matches the disease you want to look into, and you will see a search link. You can click on it and will be forwarded to the search result page and the advanced search tool where your query will be pre-populated. And now I would like to draw your attention to two important features. First is an ability to include computer structure models into your search results, which you can do by switching this CSM toggle on, and you can do search again. You see an increase in search result hit and a sizable total of 333 polymer entities or PDB sequences that would be somewhat challenging to go through manually. So that is where an ability to render your results in a non-redundant manner comes in useful. You can activate it by using a group by setting that is located in Advanced Search tool. And here I'm interested to see all the structures relevant for given UniProt reference sequence. So I added that setting, and I would like to see group summary as opposed to single representative structure. So I redo my search and immediately see a substantial tenfold reduction in terms of the number of hits. As for many protein PDB archive, the PDB archive includes multiple structures coming out as a result of different experiments. So now we have only 25 hits, and we can look through them. There are many proteins that we've seen in this list, including that are associated with the disease, including our protein of interest, which is the low-density lipoprotein receptor adapter protein 1. From here, you can navigate to the page, we call it a group summary page, which summarizes all structural data available for this particular protein. And as you can see, there is only one experimental structure and one computer structure model for a human LDL-RAP1. And what I would like to see next is how those structures are aligned to the reference sequence, which you can do by clicking on this Explore Sequence Alignment in 3D link. Upon clicking on this link, you are forwarded to a tool that presents you with in a two-sided panel. It presents you with sequence information on the left and structure information on the right. And those two panels can be explored bidirectionally, meaning that you can locate a feature of interest on the left side and visualize it in 3D and vice versa. So what's interesting is you can basically see both of the structures. One is a PDB structure, another is a CSM mapped to the reference sequence. And as you can see, there is a only short fragment that is available with experimental structure, which is mapped to this helices here. But there is a large spanning across almost half of the protein section covered by CSM, where you have high quality prediction. You can see other features. We call them features. They're positional features, which can be a point on your sequence representing a genomic variant, for example, or a modified residue or potential mutation site. And you also see features that are spanning multiple residues like domain. One example is a functional PID domain, which mediates interaction with cytoplasmic tails of the LDRR. And so you might be asking, like, if there is no experimental structure, how come there is this high quality prediction in this part of the protein? And I would like to take you back to the group summary page and show you another path of how you can explore CSM data. You can go to the structure summary page for this particular structure, and you see there are a couple of handy search links that are made available for you. You can search for proteins of a similar sequence. You can search for protein of similar structure, which I'll do right now. For example, this link brings you back to advanced search tool with, again, your input pre-populated. So what you're asking now to do is find all the structures on our known to our CSB.org website that are similar in shape to my query structure. And you can see there is a way to control whether you want your search to be more relaxed or more restrictive. You can do that and see that now we have many more hits available. And from here, you can filter back to see only experimental data that is similar to your computer structure model. Among those search hits, you see a human structure for disabled homolog 2. So now what you can do is you can see how similar those two proteins actually are. And by clicking on this Align in 3D link, you will be forwarded to a tool that allows you to do that. You can examine structural alignment for this tool and see that despite the low sequence identity, they're very, very similar in structures. So let me bring you back to the slide deck where I am where I will tell you a little bit more about this disabled homolog 2 protein. It's a signal transduction protein that contains the same phosphotyrosine binding domain, and like LDL-RAP1, it binds to and receives signals from members of the low-density lipoprotein receptor or LDRR family. So with this exercise, I demonstrated that our CSB.org offers seamless access to a suite of very powerful tools designed to explore structure data and computer structure models that are available on our CSB.org and provided alongside experimental structures help to bridge gaps in structural data exploration for proteins with low experimental coverage. That concludes a my case study. And without further ado, I would like to introduce Sebastian Dietrich, who is a scientific software developer for the RCS PDB organization, and he will take you on a journey of exploring case study of class 2 aminoacyl-tRNA synthetase. Sebastian.
Yeah, thank you for the kind introduction, Jana. So I would like to talk about part of my PhD project that I did together with many colleagues and friends before joining the RCSB PDB in 2019. And back in the day, all of us collectively decided to work on basically a pet project dealing with aminoacyl-tRNA synthetases in addition to our respective projects, just because it's such an intriguing topic, and maybe I can share some of that excitement with you and also use it as a jumping off point to demonstrate what you can do with search tools provided by rcsb.org and how you can basically navigate our research-focused web portal and also use this to derive new knowledge from the recently integrated computed structure models. And as I said, this was like a collaboration in basically 2017, 2018. And I want to give a shout out, especially to Florian Kaiser, who was pivotal to many aspects of this project, and also my former supervisors, Dirk Laputte and Michael Schröder. So why you should care about aminoacyl-tRNA synthetases is because they are a superfamily of enzymes that address a really important process in all forms of life, and that is attach the correct amino acid to tRNA molecules so that those loaded tRNA molecules can then be consumed by the ribosome to build up nascent protein chains. So it's basically essential for the protein biosynthesis process. And again, it's really interesting for both evolutionary aspects as well as understanding the earliest stages of life, just because it's such a basic process that is shared by, again, all living organisms and is present in all living cells. And it doesn't stop there. There's not a single implementation of an aminoacyl-tRNA synthetase, but all cells contain 20, roughly speaking, implementation. So there's a dedicated aminoacyl-tRNA synthetase for every single amino acid. And as an example, you can see this two-step reaction. Here I'm using an aspartyl-tRNA synthetase as representative of what's going on. So in the first step, this enzyme will recognize aspartate or whatever the corresponding amino acid is that needs to be recognized, and also binds to ATP as kind of the energetic driver of this reaction. It covalently binds both of them, and in the second step, this activated amino acid can then be used to charge the tRNA molecule with an anticodon specific to aspartate with the proper amino acid. And then you get this product of a tRNA that is specific to aspartate and charged with an aspartate residue so that the ribosome can consume it and build up protein chains. And everything related to protein biosynthesis always has this bootstrapping paradox that you basically need the whole machinery present to build the machinery itself. But I would argue that aminoacyl-tRNA synthetase is at another level of complexity to this, because they don't just have the self-referential characteristics, but they are what implements the genetic code at the end of the day. So those are the enzymes that make this connection from certain codons or anticodons or nucleotide triplets, whatever you want to call it, to the corresponding amino acids. So we can also learn about the origin of the genetic code potentially, as well as understand why organisms use the set of amino acids that we see today as being relevant. And to provide some further context, so if you consider the 20 amino acids that we usually care about, you can group them by their physicochemical properties, and the interesting thing for aminoacyl-tRNA synthetases is that, again, there's a dedicated implementation for every single amino acid, and they tend to be really dissimilar just because we are looking at billions of years of evolution. And you can find these pairs and try to figure out what their sequence identity is, and it usually hovers around 25%. So there's some signal there, but not a whole lot of signal. And structurally speaking, they also tend to be quite dissimilar. So over the course of evolution, certain organisms might recruit additional domains, which leads to a different overall global structure for these enzymes. But scientists have been able to identify two classes of aminoacyl-tRNA synthetases, class 1 and class 2. And there's like a really neat separation of amino acids between those two classes. So class 1 handles roughly 10, class 2 handles roughly 10, and also in terms of physicochemical properties, you get like a really nice divide of having acids in both classes, having aromatic amino acids in both classes. So you can't say that one class life couldn't function or is not viable with just one set of amino acids handled by class 1 or class 2, but you have another layer of this intertwinedness. And so again, sequentially, they are really dissimilar. They are dissimilar at structure level, but class 1 tends to be a Rosmanoid fold, whereas class 2 is pretty unique and has a anti-parallel beta sheet conformation that we don't really see in other structures whatsoever. So this seems to be a unique fold that evolved. And what we tried to do back in the day was to find some consistency in both classes. And that's an interesting proposition because they are not just dissimilar at sequence level, at structure level, but also functionally. They have like a lot of peculiarities that depending on how many cofactors they need or what's their quaternary structure. So there are like many differences between class 1 and class 2, and whatever characteristic for a protein structure you can imagine, most likely class 1 and class 2 differ, despite basically realizing the same biological function of charging tRNA molecules with their proper amino acid. And so our hypothesis back in the day was, well, okay, everything is different, but both need ATP as an energetic driver of this reaction. So surely ATP recognition in both classes would have some common feature that we can identify. And we published in 2018. And at that point, we were the first group who did like an archive-wide analysis. So we took everything that was available on rcsb.org back in the day and filtered for everything that is an aminoacyl-tRNA synthetase. And we got a pretty nice data set of like roughly 1,000 structures. And we got this nice divide between, roughly speaking, 50-50 between class 1 and class 2. And we also had data for every single amino acid that we cared about. So pretty promising start. And now we were trying to find consistent interaction patterns with respect to this ATP ligand in the binding site of all these structures if ATP is present as a ligand, and to figure out some consistent pattern of non-covalent interactions that we might be able to identify. And fairly soon, we identified one consistent pattern in class 2 of 2 arginine residues that surround the ATP ligand. And these arginines are highly conserved at sequence level, and also their interactions, how they bind ATP, this is highly conserved. So here you can see this guanidinium group perform or participate in pi cation interactions with this aromatic ring system. And the other arginine residue uses its side chain to form these salt bridge interactions with the phosphate group of the ATP derivative. So this is a fairly self-explanatory motive, and we saw this being present in pretty much all class 2 aminoacyl-tRNA synthetase structures. So this is like a pattern specific to class 2. And we called them the arginine tweezers. And again, you can see them clearly at sequence level if you know where to look for them. And now we try to identify a similar pattern in class 1 aminoacyl-tRNA synthetases, and we're really scrambling to identify anything that would stand out in the same fashion. And at some point realize that basically the pattern is that there isn't any consistent pattern really going on in class 1, but rather we see a pretty unique kind of ligand binding interactions where there's no sequence conservation, strictly speaking. And that's because the side chain of those amino acids doesn't really matter. So you can see side chains pointing away in this rendering. And rather, there are really generic hydrogen bonds formed by the backbone atoms of those amino acids. And virtually any amino acid can form them. Maybe proline would be a bit weird, but again, generally speaking, all amino acids are welcome here and can participate in those generic hydrogen bonds formed by the backbone atoms of the corresponding amino acids. And with that in mind, I think it would be nice to basically revisit the study and to see how the PDB archive changed on one hand, because in 2018, we had like roughly speaking half of the data, and in the 6 years since then, roughly 100,000 new PDB structures were deposited, and some of them are aminoacyl-tRNA synthetases, so we got more data. And also we now integrated 1 million computed structure models from various model organisms as well as genomes with global health implication, which means we got many, many genomes. And I told you that all of them contain more or less 20 aminoacyl-tRNA synthetase implementations. So we got like a lot of data to analyze and to search in. And that's what I would like to do for the second part of my demonstration, to basically show you how you can use the rcsb.org web portal to answer these questions and to find similar structures for class 2 aminoacyl-tRNA synthetases. And again, I think, as Jana pointed out, one of the best jumping off points to do that is usually a structure summary page. So here I just went back to this initial example of an aspartyl-tRNA synthetase and picked one structure as kind of a representative for everything going on in class 2. And now we can use different search services to find similar structures. And my goal would be to find everything in class 2, everything that is a aminoacyl-tRNA synthetase. And we can try to do that using a sequence search. We can try to do that using a global structure search that looks for similar shapes in terms of individual protein chains and find chains that look highly similar to that. And last but not least, there's also a tool that allows you to search for structure motifs. So you don't care about any global similarity, and you just define a local binding site motif. And the arginine tweezers are perfect for that. So we can define a motif on this pair of arginine residues and then run a query that gives us access to all other occurrences in the PDB archive and the computed structure models that contain 2 arginine residues in this characteristic arrangement. So with like a certain distance from one another and a pretty well-defined relative angle between their side chains. And long story short is that in this particular case for aminoacyl-tRNA synthetases, just because we are looking at billions of years of evolution, a sequence search doesn't give you that many results. So in our study in 2018, we saw like roughly 500 structures in class 2, but a sequence search will only give you access to 20 experimental structures. And the same is true for global structure search, which only gives you 18 examples, just because sequentially they are too dissimilar, and also at a structure level, a global structure level, these structures recruited additional domains so that maybe a global structure search can't pick up on the signal. But I want to show you that the structure motive search in this particular case is a really helpful tool. And with that said, I think it's best to switch over to looking how you would use the RCSB.org web portal. So again, I think one of the best ways to approach this is to go to a structure summary page, and then Jana already pointed to this section, which is extremely helpful if you want to launch queries in a really convenient fashion. So you can find similar protein chains to this aspartyl-tRNA synthetase chain, either by sequence similarity at varying thresholds, or by looking for chains that share global structural similarity. And in terms of a sequence search, if you follow this link, the website will populate the search panel with your sequence. You can also search for custom sequences. It will fill in all the parameters, and you get this result set where you can now investigate those 20 chains that we found that are reasonably similar in terms of sequence similarity. So first we see the entity that we used to define this query. So nothing exciting here, 100% sequence identity. But as you scroll down in this result set, hits get more and more dissimilar. And maybe it's best to look, for example, at this structure, which gives you this nice alignment overview. So here you can see that your query aligns pretty well with this particular entity. So you get an alignment over the whole length. You have some gaps in there. Sequence identity already dropped to 40%. So yeah, more and more interesting and distant hits. And the last two hits, they don't even align over the whole length, but you only get this N-terminal match where only the beginning of your sequence matches, and here you got some C-terminal match, but not over the whole length anymore. And I just want to point out that we defined this search using an aspartyl-tRNA synthetase, and this one hit is an asparagine-tRNA synthetase. So it's also a class 2 aminoacyl-tRNA synthetase, but we were able to at least jump from aspartate to asparagine for one structure. So there's something here, but not the most exciting results set, generally speaking, why I also will skip like looking at the computed structure models that we'll find for this particular example. And if you use the same strategy but launch a global structure similarity search, something similar happens. So we define a search referencing a structure that we use to, where we looked at the structure summary page, and we say, okay, for this chain, find all other chains that look similar. And overall, the shape of this result set is the same. So again, you find your original structure first, and then you got many, many more hits. And at the very end, you can see another example of an asparagine tRNA synthetase, or actually a tRNA molecule, which is ambiguous between aspartate and asparagine. And one nice feature is, so previously had this sequence alignment overview, and now you get this button to align in 3D, which will take you to our structure alignment app, where you can align pairs of structure, or even multiples to one reference structure. So we can align this chain and this chain, and then interrogate it in 3D. And you clearly see that structurally these are highly similar, especially in this core domain that we truly care about, where the ATP is bound. But also overall, this is a meaningful hit. And if you look at the TM score, it's, it implies that this is the same fold, which is obviously true. But again, this is not the best way to find really interesting class 2 aminoacyl-tRNA synthetases in this example. And to approach it from another angle, we can define a structure motive query, which would just search for those two arginine residues that bind at the ATP ligand. And we could, for example, form the hypothesis that this might be a generic ATP binding motive. So maybe other proteins have different folds, but they evolve the same pattern of two arginine residues. to interact with ATP, and many proteins bind ATP. So maybe we find some signal, or we find some other unrelated fold that also contains two arginine residues and binds ATP. And one way to launch such a query would be to follow this Explore in 3D link, which takes you to our Moster 3D viewer to visualize everything in 3D, and you get basically different things that you might care about, like electron density if it's available, or validation of the structure. But what I want to do is to look at the ligand interactions, and if you follow this link, you get this view that zooms in on this AMP-derived ligand, and now with some eagle-eyed skill, you can select those two arginines that I initially told you about. So here you can see those p-cation interactions, and here's the second arginine that interacts with this phosphate group. And this will populate the structure motif search panel, and then you can click Submit Search to launch this query and to find all other occurrences of class 2 aminoacyl-tRNA synthetases. And again, this advanced search query builder gets populated with the data that you're interested in, and you get 1,600 results, which is a meaningful number. So initially we had like 500 structures, roughly speaking, in class 2 aminoacyl-tRNA synthetases when we conducted this study in 2018. And now you find more or less 1,000 more chains that contain this arginine tracer motif, which kind of checks out with how the PDB archive grew over time. And again, the first hit tends to be not really that interesting. So this is just the structure that we use to define our query. But the second structure is already pretty interesting. So this is not a clear tRNA synthetase anymore. And that's because we are just looking for pairs of arginines in this characteristic arrangement, but we don't really care if it's an aminoacyl-tRNA synthetase or if it isn't. We're just looking for for arginine pairs that potentially bind ATP as a ligand. And again, you get those align in 3D buttons, and you can use them to like interrogate this structure and to see what's going on. And if you follow this link, this view will open, and here you can see the arginine tracer motif from the original structure that we used to define this query, and also the second structure, like the hit that was identified by the algorithm. And you can see that in this core domain, in terms of structure, these are highly similar. So this is a really nice alignment. And again, the alignment is not based on any global similarity, but just the two pairs of arginine residues get aligned. And in terms of our question whether this is an ATP binding motif, we can see that it is also true that for the structure that we use to define our query, there's ATP present. And also for this hit that we identified, we can also see another AMP charged with an amino acid. And again, if we go back to this corresponding structure summary page and read up in this publication, we will see that this is a paralog of a lysyl-tRNA synthetase, which basically means that, yeah, it's related to a class 2 aminoacyl-tRNA synthetase, but after a gene duplication event, this copy was able to evolve into some potentially different functionality. And it's not really understood what this functionality is. We only know that it involves ATP binding and that it's evolutionarily related to the class 2 aminoacyl-tRNA synthetase fold. And moving on, like another angle would be to see, what can we find out in the data set of computed structure models? And again, as Stephen and Jana emphasized, to do that, you want to switch this toggle to its science state, which means that computed structure models will be included in your search. And I also added one additional criterion, which is called structure determination methodology, which allows you to filter between computationally predicted models and experimentally determined structures. So here I'm selecting computational, so we only see predictions from AlphaFold DB and from the model archive repository. And we can now investigate this result set. So most of the time you see pretty classic class 2 aminoacyl-tRNA synthetases, but for example, this is a histidine tRNA synthetase, so it's not the original aspartate one that we used to define our query. And actually, most on page 1 is a histidine tRNA synthetase, but as you navigate your result set, and even at the very tail of this result set, you can find different class 2 aminoacyl-tRNA synthetases. And one thing I would like to focus on is just by chance, the first hit that we identify is also not a clear-cut case of an aminoacyl-tRNA synthetase. And again, we can leverage this align in 3D button to see what's going on and to basically zoom in on this alignment of the arginine tracer motif. And again, you can see that in terms of this core domain, the fold matches really well. The arginine tracers, as predicted by AlphaFold 2, match remarkably well. And you can only see a single AMP-derived ligand in this binding site coming from the experimentally determined structure, just because AlphaFold 2 currently doesn't include any ligands. But you can still make the case that at least there's a binding site here, and you got space to bind ATP. The overall fold in this core domain seems to be the same, and also the arginine tracers are present in both, and we know them to be an ATP binding motif, which doesn't seem to occur in unrelated folds, but it's present in this class 2 aminoacyl-tRNA synthetase fold with its characteristic beta sheet or anti-parallel beta sheet conformation. So maybe there's some signal here. And again, if you then read up on this particular enzyme, you will see that it contains domains which were most likely recruited from class 2 aminoacyl-tRNA synthetases. And with that out of the way, I would like to summarize that for the class 2 aminoacyl-tRNA synthetases, we can use different search strategies. And in this particular case, sequence search and global structure similarity searches don't find anything really that interesting. So we can identify aspartyl tRNA synthetases, but nothing more generic in class 2. But in this particular case, the structure motif search tool identifies other class 2 aminoacyl-tRNA synthetases just because the arginine tracer motif is a signature motif for this family of enzymes. And to generalize some more, ACSB.org gives you access to like this wide range of search tools and strategies, and makes it quite easy to navigate enormous result sets. And I think especially for computed structure models, this is really important just because we got so much data now, and you need powerful tools to make sense of them and to compare them. And I think it's really important that you don't just use a single tool, but you get access to the suite of tools and baked in visualization that you can align everything in 3D like a single click. So I think that's a pretty convenient way of investigating computed structure models. And I also want to emphasize that we integrated 1 million computed structure models from AlphaFold DB, as well as the model archive repository, where scientists can submit their own predictions using whatever algorithm performs well enough to make it into that selection. And AlphaFold DB mainly contains model organisms, including predictions for the human genome, but also genomes with global health implication, whereas Model Archive, for example, contains genomes that are relevant for combating climate change. So here you can see some related resources, and big shout out to Brinda Wallad and Model Civ, which is basically the bedrock of this integration, and which made it really easy for us to integrate computed structure models from AlphaFold as well as Model Archive. And with that said, I would like to draw your attention to another upcoming event, another virtual webinar that will answer the question, which experimental structures should I rely on? And this will happen in 2 weeks on May 14th at 2 p.m. Eastern and 11 a.m. Pacific time. And I also want to emphasize that there will be an exit survey, which is 10 questions, takes like 2 to 3 minutes, and it really helps us to organize these events and to understand what you as an audience are interested in, and also how we can improve these events. And by filling this survey out, you will be eligible for a participation certificate. And last but not least, I want to thank you as an audience and also all RCSB PDB team members, because all of this is a team effort. And especially I want to give a shout out to Shamara and Michael and Christine, who were organizing this event behind the scenes. And with that, I would like to open the virtual floor for questions. So thank you very much.
Thank you, Sebastian. Yeah, now that we covered the main points, let's open the floor for the questions. We set aside roughly 15 minutes for Q&A. And today with us are also Jose Duarte, Brinda Valet, and Joan Segura Mora, who are deeply involved in development of the CSB.org platform. We welcome questions from all perspectives, so feel free to raise your hand or post your questions in Q&A box. And with that, I'd like to invite the first question that I'll pick from the Q&A box, which is how is the similarity of structures determined? What determines the strict versus relaxed cutoff? Excellent questions, and I hand it to Jose to answer.
Yeah, thank you, Jana. So maybe can I share my screen and just to show a couple of things, pointers in the text. So let me share this quickly. Yeah, just to show that we have help pages in our website, in our CSB.org. So if you go up here, you'll see help about search. One of them is, that's what I navigated here, is a structure similarity search. And so this tells you everything about how it works. Just very briefly, I'll give you a like few sentences explanation. It's complicated, but I can give you a little overview. It's all based in in-house research done and published in this paper by Gusenko et al. In 2020. Very briefly, I'll open the paper and one figure in the paper that's important. It's all based in a mathematical tool called 3D Zernike moments that sort of describe as a volume, like this hemoglobin tetramer that you see here. You can describe it with summing certain polynomials that more and more gradually approximate the volume. Based on this, you can get essentially a vector to describe the volume, and that gives us a way to very quickly compare the volumes, be it of chains or assemblies or any kind of assembly in the PDB. That's how the system works, and there's a couple of caveats. The most important one is that it's comparing global volumes, so global shapes only, if you happen to have a certain domain that is in common between two structures, but otherwise the structures are different, then it won't be matching. So it will be matching the global shape only. And then to the question of the two different cutoffs, it's a matter of based on the this method was trained against some data set. based on that training, there's two different cutoffs. That's explained somewhere here too. Relaxed and strict, and they will be essentially the strict one will try to give you relevant hits without having too many false positives, while the relaxed one will be more relaxed and you can at the expense of having more false positives in the results. So definitely when you look at the results, try to look at the scores, which give you an idea of more or less how good this structure is. And as Jana and Sebastian showed, also you can always open the link when you are doing chain search. I'll do a quick demo now again. Be sure that you click on the alignment, because that will give you the full view, and you can be sure that so for instance, you can align with this button here, and that you can be sure that it's a good hit or not, and you will also understand, you'll get the metrics in RMST and sequence identity, etc. Okay, I hope that answers that question.
Thank you, Jose. And I'll move on to the next question from the chat. How do you clean the data obtained from structure motif search, because it is not looking for global structure, rather only particular segment?
That's a really good question. So like back in the day, what we used to create this data set was looking at enzyme classification numbers, for example, and other annotations that are present for protein chains, because especially for like paralogs with unknown function, it's really difficult to gauge whether they are, functionally relevant or not. So I would like to reiterate this point of the annotations tab, where you get also functional annotations from varying resources. But yeah, it's a really difficult question, and usually you need to do it on a case-by-case basis. And maybe another thing that can help you with this is the refinements panel, which you can use to navigate your results set. So maybe you want to focus on, human proteins, because maybe those are better understood to answer that question. But yeah, unfortunately, I can't give you like a clear answer. So that's kind of the advantage of a structure motif search, that you don't care about any global similarity and only look for this local arrangement. But it can also be a drawback that, two arginine residues could just happen to be oriented like that by chance. And yeah, another aspect is that you can also refine your motifs. So you could try to add additional residues and make it more specific, or to arrive at something that describes a binding site with more information, which will effectively reduce the rate of false positive hits or of not biologically functional proteins in your result set, depending on how you define your query. And maybe there's another question related to that, how you could programmatically use these tools. So you're not required to use Monster to define these motifs. So you can just use this form to say what are the residues that you're interested in, and you can add residues to this. And once you settled on a motif, everything is backed by public APIs. So there's also a programmatic way to directly launch those queries, to manipulate those queries in a more flexible way if you know what you're doing, and to approach this problem from that angle.
Thank you, Sebastian. Moving to the next question. For computer structure models, which sequences are being used UniProt? Brenda?
Yeah. Hi. So for computer structure models, in the model CIF file, we capture the sequence of what's being modeled. For AlphaFold models, for the most part, including the ones that are split, it corresponds to what's in UniProt. but not necessarily. It can deviate, and if it does deviate, then that's captured in the model CIF file, and that's what we use in our backend. So most cases it will match with UniProt, but doesn't have to. Depends on what is being modeled and what's being captured in the model CIF file. But you'll be able to do a sequence search if you have your UniProt sequence and be able to get that information.
Thank you, Brenda. That answers the question. And moving on, will the CSMs be updated as the prediction tools like AlphaFold2 get more advanced and refined? And I'll hand it back to Jose.
Yeah. So yes, the field is moving very quickly, and we are trying to keep up with what's happening. At the moment, we have integrated around 1 million models from AlphaFold-DB and a few from the model archive. And we plan to, with this current integration that we have, we plan to keep updated with whatever releases the AlphaFold-DB makes or model archive. So whenever these models are updated with fixes or with better predictions. And longer term, we are planning to integrate more models, and we will then, basically, wherever we integrate those models from, we will basically keep up with the releases that the resource makes. So, yeah, I hope that answers the question.
Thank you. And next one is up. I find that you use the term fold in a very loose way. Does it mean same arrangement of secondary structure elements, or that they are in the same sequence along the polypeptide chain? In other words, same topology. And I'll hand it to Joan to cover that.
Hi, thank you, Jana. Yeah, I think that's a good point. Sometimes the term fold can have different meanings. But in this case, what we are describing is that it's not only the same elements of secondary structure, but they also have the same arrangement in the 3D space. So you can think about that both proteins are in close when we compare the superposition of the atoms. Now, what would be having a low root mean square deviation or a high TM score?
All right. And next question is coming from the chat. How one can select PDB IDs for a particular protein target based on resolution or performing molecular docking? Okay.
Okay, so I mean, I'll see what I understand there in that question is if you know a certain, I mean, you know you're interested in a certain protein based on sequence or perhaps structure similarity, and you have several PDB-IDs to choose from. How do you select the ones that is more well, it has higher resolution? That's what I'm understanding from the question. There's a few ways. I can show perhaps, okay, I'll share my screen again. I can show one. So let's say that I was, let's go back to my search, which was for hemoglobin. So I did structure similarity search for chains that are similar to one of the alpha hemoglobin in the hemoglobin tetramer, and I'm getting all these results. So one thing I can do in this result set is going to the refinements panel and then do something like this. So, well, I'm interested in high resolutions only, so I can go here and refine my search and just show the ones that are high resolution only. That's one thing that I can do. You won't see the resolution here, so you will have to go into the structure summary pages and look at your results. Or you can create a report. Some of these reports do have resolutions. There's also other ways. Maybe I can show, okay, if I go back to the result set as it was, the raw result set, I can do something like grouping by sequence clusters, so by sequence identity, let's say at 50%, if you are interested in sort of a range of structures that are related in 50% sequence identity, I can group like this, and once I group like this, I will show the results of representatives. So I click on search again, and I'm looking at results grouped by 50% sequence similarity. So now they are redundantly reduced, and there's only 71 representatives out of the whole more than 1,000 that we had before. And in here, the representative is automatically selected as a good resolution one, but I can even do more. I can, instead of representatives, I can show as groups, and that gives me links to the groups. So for instance, hemoglobin subunit beta, I can go to the group, and I get for this group, I get all the members in this panel, so I can navigate through them, but I get like statistics for all of them. And one of the statistics I get is resolution. So I can click here. So similarly to the drill down panel, I can click here, and I can then get only those that are high resolution, and then those are shown here, so I can essentially navigate through them and then try to open the structure summary page and further dig into it. Yeah, hopefully that gives a few different ways to navigate your way around.
Thank you. We are past reserved time for the webinar, but let's take a few more last questions. And one of them, many thanks for this very useful webinar. Well, thanks to you for nice comments. My question is about if there is any way to identify or recognize the type of the interaction between amino acids, especially if when two amino acids are not of the same type, for example, hydrophobic with charged one or polar with charged one. Okay, Sebastian?
So I think a good way to approach this is this ligand interaction feature that I showed, which allows you to, for example, focus in on one ligand. But in Monster, you can actually click on any residue, and you will get this interaction network, or you will see the non-covalent interactions that are formed with its neighbors, which gives you a pretty good overview over hydrogen bonding patterns, but may not include like more specific things like hydrophobic interactions. But there are also dedicated tools for that available on the internet to run that analysis. And maybe another angle to answer this is the structure motif search doesn't care about any actual interactions that are formed between the amino acids and some ligand, or it doesn't even care if there's a ligand present whatsoever. And we have tools to search for specific ligands, but there's nothing really available yet that would connect certain interactions between ligands and the protein and allow you to search for specific hydrogen bonding patterns or patterns of hydrophobic interactions or anything along those lines. So, yeah.
Excellent. Thank you, Sebastian. And one last question that we'll cover today. Can unknown sequences help us in deriving a particular protein by similarity search If so, how do I progress? John?
Yeah, thank you, Jana. So I understand that in this situation, when you don't know the sequence, but I guess you are asking in the reverse problem where you have the position of the main chain and you want to know what sequence could fall in that particular structure. You can always use our advanced search and send your PDB with the main chain atoms, and our search will provide you with the collection of potential structures that are similar to yours.
Thank you, John. Well, with that, we will conclude for today, and I want to thank everyone for your time and your involvement. Your questions helped us to deeper into the topics that we covered today, and I hope this was useful. Please fill in the exit survey, which will help us understand if this type of events are useful and you want to have more of them. And thanks again to everyone.